All right, today we are going to focus on the nucleus, ribosomes, and the endomembrane system. The first two of these, we put together an LBL worksheet for you to go through to learn some of the basics, and we're going to use this material to answer some questions in class. And then we'll move on to the endomembrane system, which consists of the nuclear membrane, which is contiguous with the endoplasmic reticulum membrane. And then we'll go on to the Golgi and end up with the lysosomes that come off of the Golgi. Okay, the nucleus, as you learned from the LBL, contains the genetic material, right? We have in the nucleus, we have the chromatin in a non-dividing cell, which is the DNA that's not highly compacted, along with its proteins. And this is all surrounded by a double membrane. And we know from earlier lectures, right, that lots of things don't go directly across a phospholipid bilayer very well, right? Charged molecules, large molecules, and things have to go in and out of the nucleus. And that's accomplished by nuclear pores in the membrane. These allow transport of proteins and large macromolecules into and out of the nucleus. So here, this is the inside of the nucleus, here is the outside of the nucleus, so this is the cytoplasm, and we have these nuclear pore complexes that allow things to go from the inside of the nucleus to the cytoplasm, or from the cytoplasm into the nucleus. So there are nuclear transport signals on molecules that need to transit across the nucleus, and these transport signals govern the direction of flow through the nuclear pores. So to explore this in a little bit more detail, what I want you to do is start to fill out this table with the people around you, thinking about which of these molecules or proteins that we've talked about already have most likely a nuclear import signal, a nuclear export signal, neither or both. To answer these questions, you have to look at each one of these things and decide, where is it made in the cell, and where does it function in the cell? And then that will allow you to make a good guess at whether it needs a nuclear import, export, neither or both signals. So go ahead and talk to your neighbors. This is in your notes, so fill in this table as best you can. And then we'll talk about why. All right, let's go ahead and start at the top of this list now. Sodium, potassium, what type of molecule is it? Okay, some people, the answer was both, but what type of molecule? It's a protein, right? Okay, and so this is the membrane, and so none, import, export, or both. How many say none? You. Import, raise your hand. Export, both. Okay. The majority of you are not right. But I do need you to tell me, somebody who said both, I need to know why you said both, because that will help me understand what I haven't gotten across to you yet. So some brave person who said both, tell me your reasoning. Yes?
I would say that it is both because not only does it allow the sodium out, but it allows the potassium to be so it's importing and exporting.
Okay, so her answer was based on the fact that the sodium-potassium pump, pumps sodium and potassium across the membrane. It pumps potassium in and sodium out. Okay, that helps then. Now, so what we're talking about is nuclear import. So this is in the plasma membrane, and it's a protein, so we know that it's made in the cytoplasm Rafi-Yara area, right? And then it is transported to the plasma membrane. Was it ever inside the nucleus? No. Does it ever need to go into the nucleus? No. Okay, so now what is your vote? None. Okay, very good. Okay, so that really helps me. That was a good explanation for why. So it's, you have to think about where these are in the cell and where they're made. Okay, mRNA then. Does it need none, import, export? So where is it made? Made in the nucleus. Where does it function? In the cytoplasm. So what type of signal does it need? Export. Very good. Okay. And it doesn't need to go back into the nucleus. Okay. DNA. Import, none, import, export, or both? None, right? So it is made in the nucleus. It's replicated in the nucleus. It functions to actually direct synthesis of mRNA in the nucleus. And it doesn't function outside the nucleus. Okay. Microtubules. What type of protein, what type of molecule is it? Protein, yeah. So where is it made? The cytoplasm, and it's a cytoplasmic protein. Okay, and so does it ever go into the nucleus? No, it doesn't need to be in the nucleus. Okay, so none. DNA binding proteins. Where are all proteins made? Okay, out in the cytoplasm. And so where is something that's going to bind DNA? Where is it going to have to go? Into the nucleus. That's exactly right. So it has import signals. I just learned and thought about it more carefully that, in fact, there are cases where DNA binding proteins are actually exported out as well as a mechanism of regulation. So this could actually be both. Okay, and finally, proteins that assemble with rRNA to form ribosome subunits. This isn't something I talked about in lecture, but it was covered in your LDL. Where are ribosome subunits formed? In the nucleolus, which is inside the nucleus, right? So we have proteins. Where are they made? Cytoplasm. Then they get assembled in the nucleus, the nucleolus in the nucleus. And then where do the ribosomes function? Outside. So what do they have to have? Both. They have to be able, the proteins have to be able to get inside so that they can assemble to form the ribosomal subunits, and then the subunits have to get exported to the cytoplasm where then they can join up with mRNAs and then each other and form functional ribosomes. Okay, so this is a type of exercise that helps you really figure out these molecules that we've talked about so far, that you'll know a little bit more about them because you've interrogated them from a lot of different angles now. Where they are in the cell, where they're made, and what kind of signals they might have. Okay, one of your quiz questions that many people got wrong was, if you unexpectedly found actin filaments accumulating in the nucleus of a cell, which of the following components is most likely to be damaged? And the answer was nuclear pores, and that's because actin filaments are generally found out in the cytoplasm, and if they end up inside the cell, that means that there is, inside the nucleus rather, that means that the gatekeeping function of these pores is not very effective, and that's a possible explanation. And none of the other explanations that you were given as possible answer choices were true. Okay, nuclear pores are complex structures that are made up of multiple proteins, in fact almost 30. Here is a diagram of what these might look like. Here's the nuclear side and the cytoplasmic side, and things are going across this nuclear membrane, which is shown here as a double membrane, through this nuclear pore, and this is an artist's rendition of this. And you can look on the web, you can see pictures of this, and you can actually see things going across the nuclear membrane, and it's quite interesting to actually see how these function. So in post-mitotic cells, that means cells that have finished dividing, some nuclear pore proteins are present for the rest of the life of that cell. So there are cells in our bodies that don't divide much, if at all, after birth. So for instance, the neurons in our brains, most of them were born before, or at the time you were born. before or at the time you were born. And so in my brain, I have lots of neurons that have nuclear core proteins that have been around for a very long time. And in fact, just like my skin, which is not as smooth as it used to be, nuclear pores also age, and the function, and their function is not as, they don't function as well in the older cells. Okay, so nuclei from young and old cells were interrogated for tubulin. How much tubulin is around, right? This is cytoplasmic protein. And D'Angelo's group in 2009 found this interesting result, which was here we have nuclei from young cells that have only a little bit of tubulin around, just right around the edge, whereas nuclei from old cells, many of them had tubulin all through the nucleus. So the most likely explanation for this is I'm going to close the window. Okay, very good. 81% of you are correct. That the most likely explanation is old cells have leaky nuclear pores. However, it could be true that tubulin in old cells have nuclear import tags, because we see lots of tubulin in there. Not very likely, though, because as cells age, you don't often see a gain of function like this, right? What you would need to be able to do is say, okay, as the cell ages, then it's going to get more efficient, or actually not more efficient, it's going to start doing a new function, which is to add nuclear import signals to tubulin, which is less likely than losing the function that you had as a younger cell. So it's the most likely explanation is always something breaks rather than you actually make something new. But that one is a logical answer. It could possibly be true. In the same way, tubulin in young cells could have nuclear export tags. Again, not very, very unlikely, because normally they wouldn't be in the nucleus, so they would have to have an import tag and then an export tag. So those are both very unlikely, and that's why this is the most likely. And, in fact, true. Oops, okay. Now, moving on to another question that you had on your LDL. Synthesis of all proteins begins on free ribosomes. True or false? Okay, most of you clicked in already, so go ahead and finish up. Okay, and in fact, the majority of you were incorrect on your LDL, and the majority of you are still incorrect. Okay, so the correct answer is that synthesis of all proteins begins on free ribosomes. That is true. That's the very initial stage. Remember the cartoon that I said, look at this cartoon drawing of synthesis of a secreted protein in the LBL, and write out each of these steps in your own words. Where did synthesis start? On a free ribosome, right, in that picture. Okay. This is an important point, and so I'm going to go take a step backwards, and actually we're going to review where the RNA is made and where the protein is made, and then we'll get to the ribosome. Okay, here is a nucleus in our cell. Remember, it has the chromatin, which is the lightly condensed or decondensed DNA. DNA is used to make mRNA, and that all happens in the nucleus. And that DNA process of making mRNA from DNA is called transcription. Okay, that mRNA then is exported out what structure? Nuclear pore into the cytoplasm, And then it initially associates with a free ribosome, and in that process we have mRNA, then direct synthesis of a protein, and that's called translation. So you have to know these terms. DNA to make RNA is transcription, mRNA to make protein is translation, and you'll go over this in great detail when you get to Dr. Edginger's part of the lecture. Okay, so now we're out here, and what I want to do then is go through and complete this flow chart to help you understand how this process occurs. So you have a partially complete flow chart in your notes, so you can complete this with me. All right, so our mRNA comes out of the nuclear pore into the cytoplasm, and what the mRNA does is it actually binds to a ribosome. It actually binds to one subunit of the ribosome. Does anybody know which one? Binds to the small subunit of the ribosome. Then that attracts a large subunit, and then synthesis can start. So free ribosome is attached to mRNA. Protein synthesis begins. Then you can go through this flow chart and ask a series of questions. When protein synthesis begins, you can ask, is there a signal peptide on the growing polypeptide? If the answer is yes, then what happens is, I have to go through this part first, sorry. If the answer is no, then synthesis continues on the free ribosome, and this makes a cytoplasmic protein that's released into the cytoplasm. So cytoplasmic proteins, their synthesis starts on a free ribosome. It is completed on a free ribosome. Then the protein is released into the cytoplasm where it functions. And so what cytoplasmic proteins do we know about already in this class? Yes? Motor proteins. So kinesin, dynein, myosin. How about cytoskeletal proteins? What proteins are those? Yes. Carrier proteins. So carrier proteins are an important type of protein we've talked about, but they are actually membrane proteins. So we have tubulin that makes up microtubules. We have actin that makes up actin filaments, right? So those are all cytoplasmic proteins. Okay, so they're made on free ribosomes, and their complete synthesis, they start on free ribosomes and they finish on free ribosomes. Okay, if they have a signal peptide, which is what I started with before, yes, then what happens is a signal recognition particle. Anybody remember this from their LBL?
Yes.
Yeah, a signal recognition particle attaches to this and halts synthesis. And then the ribosome binds to the rough ER. Okay, this signal peptide is on two types of proteins. It's on proteins that are going to be membrane proteins and proteins that are going to be secreted. So if the mRNA encodes a secreted protein, then what happens is the protein is synthesized. So now we have our ribosome sitting on the endoplasmic reticulum, and it's synthesizing the protein, and the protein enters the ER lumen, so the space in between the ER membranes. It's synthesized directly into the lumen during synthesis, and then it's going to get packaged in a vesicle made from the lipid bilayer of the ER and transported to the Golgi for further processing. If it doesn't encode a secreted protein, it encodes a transmembrane protein or any type of membrane protein. And these proteins are inserted into the rough ER membrane during synthesis. And so these are actually threaded directly into the ER membrane, and they may be threaded in once, twice, multiple times across the membrane. And then these are transported to the Golgi by making a vesicle from the membrane that contains these proteins. That vesicle is then transported to the Golgi, where it's then further processed. Okay? So cytoplasmic proteins start out on free ribosomes, end up on on free ribosomes. Transmembrane, or membrane proteins and secreted proteins start out on free ribosomes and finish synthesis on the bound ribosomes. Okay, insulin is a small protein made and released into the bloodstream by pancreatic cells. A critical step in insulin production is synthesis on, and you can talk to each other. Okay, click in if you haven't. Okay, very good. The majority of you did get the answer right, which is B. Okay, we know that it's going to be synthesized on, it's complete. It's going to be, the synthesis is going to be completed on a bound ribosome because we know that this is a protein that's made inside a cell and it's released into the bloodstream, so it must be a secreted protein. And so we know secreted proteins are made on bound ribosomes. It's a bound ribosome and translocation of protein into the rough PR lumen. That's what's shown here. So this is the diagram you have for your LBL. And this is the mRNA is associated with a free ribosome. A signal recognition particle attaches to the signal peptide, attaches the ribosome to the endoplasmic reticulum, and then the protein is synthesized into the lumen of the endoplasmic reticulum. A vesicle will form around this, and this will get transported to the Golgi. Okay? And so if it was a transmembrane protein, it would get inserted here, and then it would go in and out of this membrane once or more times. And it is not, synthesis is not completed on a free ribosome. It starts there, but the packaging begins in the endoplasmic reticulum, and then it's moved to the Golgi. Okay, so I have a short demonstration that I need one person to help me with to hold something for my ribosome. Any volunteers? Yes, right there. Okay, well, she's coming down. Actually, your name is Michelle. Very good. If you could stand over here and hold this. I have two things here. I have, this would be what part of my ribosome? Small subunit. And this would be? Very good, large subunit. And an mRNA comes out of the nucleus and binds to the small subunit. That then allows it to bind to the, allows it to bind to the large subunit. And then as it begins to synthesize a new protein, we see that it has a signal sequence on it. So what comes along? Signal recognition particle, stops synthesis, and then what happens to this? Binds to the rough ER, and so where is the rough ER? Okay, binds right to the rough ER, exactly. And then it continues synthesis, and it goes where? Into the lumen of the ER, exactly. Okay, so now this is finished. This dissociates, the mRNA goes off, and now another mRNA comes out of the nucleus, and this time it binds to a different mRNA. That mRNA finds a large subunit, and then, okay, my very low-tech version here, we're going to make another protein, and as we read through this mRNA, this protein does not have a signal sequence. So does this go to the ER? No. Synthesis keeps happening, and it's released into cytoplasm. Very good. So not into the lumen, into the cytoplasm. Thank you very much. Very good. Okay. into the endoplasmic reticulum in a little bit more detail. This comprises about half of all of the membrane in a cell, and it's continuous with the nuclear envelope. And it's a series of membranous tubules, so it's illustrated here, and the lumen is the internal space, so that's where proteins are synthesized into if they're going to be secreted proteins. We have two different types of endoplasmic reticulum, the smooth ER and the rough ER. Smooth ER has no ribosomes on it, and so in this picture it's illustrated up here, and the rough ER has ribosomes that have docked to it, and that's shown by these purple dots, and down in this EM, this is rough ER, where there's all these little dots on the outside, those are ribosomes, and this is the smooth ER. So all of the ribosomes that are docked to the rough ER are synthesizing either secreted or transmembrane proteins. If they're not, they dissociate. When they're finished, they dissociate, and then they're in the cytoplasm waiting for another mRNA to come in. So it's the same ribosomes, the same, a small subunit, large subunit that get together and then dock that are synthesizing both cytoplasmic proteins and transmembrane proteins and secreted proteins. It's just which mRNA they attach to. Yeah, that's a question.
On the rough ER, the proteins produced there, are they only used in the membrane around the side cell, or can they be used in any membrane to the rough ER?
So
The proteins produced in
oh, okay, good. The proteins that are produced in the rough ER, if they're, for instance, a membrane protein, and you'll see this in a second, they could end up in the membrane that is the membrane surrounding a lysosome. They could end up in the plasma membrane. They could end up in any membranous compartment in the cell. But if it's a protein that's in the membrane, it's made in the rough ER. If it's a protein that's secreted, it's made in the rough ER. If it's a cytoplasmic protein, made on free ribosomes. Okay, smooth ER is a region that, or a structure that's rich in metabolic enzymes, and in many cells, there is only a small proportion of the endoplasmic reticulum that's smooth, and most of it is rough. But in fact, there are a few cell types that have a lot of smooth ER, and those are cell types that are involved in these processes. So, synthesis of lipids. So, both smooth and rough ER make lipids, but in fact, smooth ER makes a lot of lipids that are called steroids. So if you know that you make a lot of steroids in this smooth ER, what cell type or organ do you think might have a lot of smooth ER? Muscles do, but that's not because of their steroid production. Yeah. Yeah, things that make steroid hormones, or gonads, right? That's one of the classic cases where you're making testosterone and estrogen. Those are steroids, and so there's going to be a lot. So anything that's making a lot of steroid hormones. Okay, also cells that are involved in metabolism of carbohydrates. So for example, liver cells are important in breaking down glycogen into glucose and regulating blood sugar levels, and so there's quite a bit of smooth ER in the liver. Liver cells are also important for detoxification of drugs and poisons. They modify toxic substances, often by adding functional groups that we'll talk about in the next unit, that make the substances more soluble in water, so they're more easily flushed out of the system. And calcium sequestration. Muscle cells have to regulate calcium stores really tightly. What happens when a muscle cell is suddenly flooded with calcium? What process do we know occurs? It's contraction, and what is the binding that occurs? Myosin proteins bind to actin in the presence of calcium, and then they do a power stroke, and if there's ATP around, they can then let go and cover that stroke and mediate muscle contraction. So muscle cells have to really actively regulate the levels of calcium in, so they have lots of smooth ER as well. Okay, so frequent exposure to alcohol leads to decreased sensitivity to this substance, to alcohol. And this can be a term tolerance, often term tolerance. So what alcohol-induced changes in the ER might contribute to tolerance? So think about cellular changes that could contribute to tolerance associated with chronic alcoholism. Yes? Okay, so if you have extra smooth ER, then you're going to have more enzymes that are involved in detoxifying, and so that they would potentially work faster. And in fact, that's what's happened. It happens is proliferation of smooth ER in the liver, and it increases the concentration of detoxifying enzymes, and so it occurs more rapidly. So how might, so taking this one step further, how might this contribute to tolerance to other drugs that haven't been chronically used? So somebody who's used a lot of alcohol may be tolerant to levels of a barbiturate that I might not be tolerant to. Why is that true? Yes? Exactly. So with this proliferation of smooth ER, you not only increase the concentration of enzymes that are important in detoxifying alcohol, but all the enzymes, and some of them are targeted to these other substances. So that's exactly why, because you get an increase in enzymes involved in metabolizing other detoxifying substances as well. Okay. I'm going to go over this pretty quickly because we've already talked about it. In the rough ER, we have processing of proteins formed on bound ribosomes. We have both secreted and membrane proteins are made in the rough ER. For the secreted proteins, we know they enter the luminal space, so down here, and where they fold into their correct shape, and then they're transported in vesicles to the Golgi. And we have membrane proteins that are synthesized into the membrane, so they would be synthesized like this, so they would end up in the membrane like this, and then a vesicle would be formed from that membrane and transported to the Golgi. And the rough ER can also make lipids, and that's really important. They make lots of phospholipids because they're always pinching off these pieces of membrane to form vesicles to transport the proteins that are being made to the Golgi. So the Golgi apparatus is the structure that finishes, sorts, and ships these proteins. It's a series of flattened membranous sacs as shown here. The cisternae are the internal compartments here, and the cispace is where the receiving side of the Golgi, so these are vesicles that have been made in the rough ER, and now they're moving into the Golgi. They transit through the Golgi like this, I'll show you in just a second in an animation, and they exit the trans space, and then they're exported to the rest of the cell. So ER transport vesicles fuse with the cispace, that's what I just said, and they move cis to trans. And each cisterna contains specific enzymes that are important in modifying the proteins, either the secreted proteins or the transmembrane proteins that are contained therein. And they can also modify the phospholipids by adding, for instance, carbohydrates, polysaccharides to the phospholipids, making them into glycolipids. They can also modify proteins and glycoproteins. This organelle is also capable of manufacturing some macromolecules like polysaccharides. And then when transport out of the Golgi occurs, when a vesicle is formed at the trans face, the face away from the rough ER, there are external targeting tags that are put on this membrane so it gets to the right place. And they move along microtubules. So if there is a newly synthesized sodium-potassium pump, what type of motor protein is going to move it out to the plasma membrane? Shout out. Not very sure of this. Kinesin. Kinesin is a plus N directed motor. It is moving from the inside of the cell to the outside out toward the plasma membrane because the plus ends of the microtubules are oriented toward the plasma membrane. Okay, definitely need to know that for the quiz coming up and for all of the motor proteins, which direction they move and what microtubules they move on. Okay, here's a little animation.
A secretory protein is synthesized inside the rough endoplasmic reticulum, migrates through it, and exits inside a vesicle. The vesicle travels through the cytoplasm to the Golgi apparatus. The Golgi apparatus modifies the protein. The protein is dispatched from the Golgi in a transport vesicle that then fuses with the plasma membrane, releasing the protein to the outside.
But remember, there's a motor protein that's carrying this vesicle along microtubule tracts. Final organelle is the lysosome, which is the organelle that's involved in digestion within the cell. It is basically synthesized in the rough ER, and this goes to one of your classmates' questions earlier. Membrane proteins that are enzymes that are important for actually digesting things inside the lysosome are made in the rough ER and put into the membrane. Also, membranes are made here as well, right? So the phospholipid bilayers, because the vesicle that forms in the rough ER is actually transported to the Golgi, where enzymes are activated and a pre-lysosome buds from the transphase. So here, this vesicle contains lysosomal proteins that then are going to, from the rough ER, that are going to fuse with the Golgi, and it's going to transit the Golgi and the enzymes will get modified and activated, and what exits is called a pre-lysosome. And it really, there are a couple of additional modifications that occur before it becomes a fully functioning lysosome. Okay, what we want to look at here, the last thing is the lysosome membrane. It isolates the digestive enzymes from the rest of the cell. And they have proton pumps, which must have been made in the rough ER because they're in a membrane. And they maintain a low internal pH. And so if we have a lysosome here and it has a low internal pH, our outside cytoplasm is pH 7, we put in our proton pumps. Which direction do we need to pump free hydrogen ions? Into the cell or out? Raise your hand if it's in. Raise your hand if it's out. Okay, it's about equally split. Most people don't want to say. So if you increase the concentration of free hydrogen ions, what does that do to pH? Does it make it more acidic or more basic? More acidic. The pH goes down, therefore you're going to be pumping hydrogen ions into the cell, into the lysosome, and that is going to decrease the pH, and now the enzymes are going to be able to function. Okay. So our lysosome enzymes function best at low pH, so they need to have a high free hydrogen ion concentration. They hydrolyze proteins, fats, polysaccharides, and nucleic acids. Those, we'll talk about the process of hydrolysis in section 2, unit 2 coming up. And lysosomes fuse with phagocytic vesicles, like we saw in the white blood cell, that contains a bacteria to digest it. And they can also fuse with damaged organelles, where they break down macromolecules and recycle them. So this is the recycling plant for major macromolecules. Okay, remember we have a quiz on Wednesday at the beginning of class.